So I also want to thank the organizers for giving me this opportunity to show you today my research from Dan Littman Lab in NYU to the Israeli community. So this is great. So all living creatures are continuously interacting with the environment. As humans, we interact not only with people, but we also interact with the microbes that live within us. We interact with the food we consume. We interact with pathogens that attack us from time to time. And so the immune system monitor and regulate those interactions. And while it induces inflammatory responses to pathogens, it was evolved to allow a holistic relationship with the gut microbes. And the way the immune system achieved this is by executing different effector immune responses. And those different immune responses are considered to be governed by different types of CD4 T cells, so different programs of the CD4 T cells. Pathobionts are interesting class of microbes. Usually they are not harmful, but under some circumstances, they induce inflammation. And Mok Shu, a former postdoc in Dan Littman Lab, have constructed TCR transgenic mice. And those TCR transgenic mice actually have a fixed TCR recognizing the Helicobacter, which is a pathobiont, in the context of MHCII class 2. And using those tools, it was able to show very beautifully that Helicobacter induced in wild-type mice two types of T cell responses. One is the T follicular helper cells, or in short TFH, and those are important to induce IgA response. And another program it induces is the Treg response. And this response is really important to put the brakes on inflammation. And very elegantly, he showed that when Tregs are for some reason are defective, there is expansion of inflammation and inflammatory response to Helicobacter. So how those different CD4 T cell programs are being initiated? The dogma is that an antigen-presenting cell will uptake the antigen in the gut, migrate to the lymph node, where it will create immune synapse with the T cells that have the right TCR. And this immune synapse will lead to the activation and proliferation of the T cells. And the idea is that the antigen-presenting cell will also secrete a signal that will dictate the differentiation program. And while there are many types of antigen-presenting cells, many studies that were done before established the dogma that the antigen-presenting cells that actually initiate all the different CD4 T cell programs are the classical dendritic cells. But in light of and especially it was shown that migratory CDC2s, which is a subset of classical dendritic cells, are initiate the Treg program. But in light of this diversity of antigen-presenting cells, we really wanted to revisit this question and to ask a very central question. Is there a common antigen-presenting cell that can initiate the different CD4 T cell programs independently to what it sends? Or can it be that there are actual distinct antigen-presenting cells, and each of them is specialized in the initiation of different CD4 T cell program? And I'll make a little spoiler, and I'll tell you that the take-home message of this presentation is that we found that indeed this is the case. We found that these distinct antigen-presenting cells have a complete different outcomes, and we also found we also actually challenged the dogma that classical dendritic cells are the only cells that initiate CD4 T cell programs, and I'll show you the result in a while. Okay, so our early experiment really suggested that migratory CDC2s, in agreement to in agreement to the dogma in the field, are the ones that initiate the Treg response, and we used a Cre line that is considered to be a dendritic cell specific, and we knock out MHCII class 2, so the ability of those cells to present an antigen, or chemokine receptor that is needed for the migration of those cells to the lymph node. And in those mice, we saw complete ablation of the Treg response. as I said before, in line of what people think in the field. Interestingly, I want to point out that already here, we saw something that was very interesting. So when mice lacked the ability to present antigen on classical dendritic cells, although we lost Treg response, the cells were still proliferating, but now the program was skewed into inflammation, suggesting for the first time that there are different requirements of antigen-presenting cells for the tolerogenic response and the inflammatory response. But going back into the Treg response that we try to understand what are the cells that initiate this response, something didn't make sense to us because we tried to use different kind of genetic models to delete different subset of classical dendritic cells. And although we got quite a nice depletion, we could never see any effect on Treg induction. And we started to think that maybe we're missing something here. Maybe the Cre lines that we think are targeting only dendritic cells actually target additional cell subsets. So to check that, together with the New York Genome Center, we performed CITEseq analysis of fate map antigen presenting cells. And we found that in addition to the population that we expected to find, we saw that those Cre target two additional populations. One is the innate lymphocyte type 3. And additional population we found that are being targeted is a population that was recently described and identified by our collaborator, James Gardner. And those cells were named Janus cells, and they're very, very interesting cells, and they resemble the thymic epithelial cells. So those two populations seems to be targeted by our Cre, and very interestingly, both of those populations express transcription factor ROR𝛄t very exclusively. And why am I telling you this? Because in 2015, the lab of Greg Sonnenberg that study ILC3s quite intensively wanted to understand why ILC3s have MHC class 2. And so they used the ROR𝛄t to deplete, to knock out specifically MHC class 2 in those cells. And what they saw is expansion of inflammation and Th17 cells. And the way they interpreted those results at the time was that antigen presentation of ILC3s must be required to kill and eliminate effector Th17 in the gut. But for us, this expansion of Th17 cells really resembled what we used to see when Tregs are being defective. And we started to wonder, can it be that the ROR𝛄t cells, those cells are actually important to initiate Treg response? So together with Tariq Najjar, we checked exactly that. And I hope this plot is not too hard to watch. But basically what we saw that mice that lacking MHC class 2 by ROR𝛄t completely don't have Treg initiation. Those cells will never differentiate into the Treg program that here is marked by FOXP3 expression. Okay, so I won't show you all the data, but I will just say in brief that we did extensive work on that, and we found that this ROR𝛄t antigen presenting cells need to migrate to the mesenteric lymph node in a CCR7 dependent manner. And there in the lymph node, they create immune synapse interaction with the T cells. And very interestingly, they express the integrins that are needed to activate TGF-beta, which is the major signal to induce a Treg response. Okay, so we're quite sure about the role of the ROR𝛄t at that point. But we still couldn't figure out what's going on with the migratory dendritic cells. And why did we insist so much on those cells? The reason is that we had quite a lot of evidence that we thought they should be involved. First of all, years of studies suggested migratory dendritic cells type 2 initiate the Treg response based on in vitro data of differentiation of cells, etc. Another point that I want to make is that we found that those cells, the migratory dendritic cells, present the Helicobacter peptide on MHC class 2, and we found that they are adjacent to dendritic, that they are adjacent to cells that just getting prime in the mesenteric lymph node. So we really didn't understand what's going on here. And we thought, does ROR𝛄t antigen presenting cell are solely doing the job, or can it be that they work together with the migratory CDC2s? And we came up with—
Ranit, 1 more minute, please.
Okay, and we came up with this model, and we wanted to know which one is correct. Are they needed solely, or are they working with migratory DCs? And in order to check that, we reached out to Terry Laufer, and we took very special mice that have MHC on. That means that all the cells in the body will be without MHC, except for the cells that express the Cre. And we put it with the ROR𝛄t Cre, and we ask a very simple question. Does ROR𝛄t alone, antigen presentation by ROR𝛄t, can initiate a Treg response? And I won't go into details because now I understand I don't have time, but the take-home message was that cells expressing only MHC by ROR𝛄t were able to induce a Treg response by themselves, suggesting migratory CDC2s are not having a role here. But very interestingly, we saw that when the mice were lacking antigen presentation by those migratory DCs, we lost completely the T follicular helper response. And that's why we saw all the time they present a peptide and they reside next to the prime T cells. So by that, I will just summarize in very briefly that we think there are multiple cells doing multiple jobs. We saw that the ROR𝛄t, we're not sure still if there are RC3s or Janus cells. We're working on that. We think they're doing Treg response. We think a completely different population is doing Th17 inflammatory response, and that the migratory DCs are doing TFH response. And this work, I want to thank, of course, Dan Littman, lab members that work with me, and collaborators that are mentioned here on the slide.